Okay, students, we're going to talk today about electron transport and oxidative phosphorylation. So let's just highlight where we were at. We did glycolysis and gluconeogenesis, but in glycolysis, we formed 2 NADH and 2 ATP, and then we formed from pyruvate to acetyl-CoA 2 NADH. That's because remember that it's going through twice. And then acetyl-CoA through citric acid cycle would produce 3 NADH. And then that's times 2. So I'll put that over here, sorry. And then also 1 GTP, which gets converted to ATP, but that's times 2 because it goes through twice. Okay, now what you should be noticing here is we didn't really get a lot of ATP out at this point. So as we now go through electron transport chain, we start to see where we get a huge amount of ATP generated. Now it is a very complex process, and so this is an introductory course. So as we go along, I'll just be highlighting the key things that you need to know because it gets super complicated as we move through the actual pathway itself. Okay. All right. So let's look at some of the initial highlights. So relatively little glucose is generated in glycolysis and citric acid. So we just went through that. And now we have to ask the question, what happens with those electron carriers? And so this is where we're going to start to look at that. Now, couple of really important ideas is that this part of the metabolic process is a series of oxidation reductions, loss of electrons, gain of electrons. So I really love this as an example of a biological battery. That a battery, as you recall, are chemical processes that release electrons to form a current. And that current goes to power, whether it be your computer, your phone, electronic devices. So in this case, we see a flow of current through the mitochondrial membranes. And that process then, that flow of electron or current is important because it's going to do some mechanical work. It's going to help pump what are called protons from one side to the other, and then ultimately allow to set up a process that allows for the formation of ATP. So this animation is really interesting. I really like it. I don't know if the link works well from here, but you can always cut and paste this into your Google, and it is a good animation that shows what we're going to go through. So what I would recommend is watching it before and then after the video lecture. Okay, so electrons are carried by a series of coenzymes, then they're passed through a chain of proteins, and this drives the generation of what's called a proton gradient. So all that sounds really difficult. So this is just an overview, and I'll show you some pics here in a moment. The proton gradient then runs with its concentration gradient to drive the synthesis of ATP. So we have a lot going on here. We have a whole bunch of enzymes. We have oxidation reductions. We have protons, H plus, being pumped from one side of the membrane to the other. And then you have what is perhaps one of the most amazing enzymes of all time, ATP synthase, that actually forms ATP. Okay, so we're back to our picture of our mitochondrial membrane. All the action for this will happen in the intermembrane space. So what is highlighted there is that you have your outer membrane, you have your inner membrane, and it's in that intermembrane space that we're going to be discussing a lot of. So it's really important for you to understand then the structure of our mitochondria. And then just as review, here's a list of all the different processes that occur where they occur. So the mitochondria has a lot of important processes, and then everything really except for our cytosol of the cell, which is where glycolysis occurs. Okay. One note is that since glycolysis occurs in the cytosol, and remember from glycolysis you form 2-NADH, that NADH has to get in here because it's within the mitochondria that it's going to get processed to make ATP. And so there's going to be some energy requirements in getting it through. And that's something that I'll just mention like that as a concept. At the very end of this PowerPoint, it shows you all these different shuttles for getting it in there. I won't ask specific questions on those, but I do want you to be aware that just like we talked about so much is that the membranes are selectively permeable. So NADH needs help to get through. Okay, so I really love this picture. So I'm going to put a little heart. And it's one that I think is really important. So it's really showing all the different amazing proteins that are involved in oxidative phosphorylation. It's also showing that it takes place on the inner mitochondrial membrane. So here is the matrix, the middle part of that mitochondria, the inner part of the mitochondria. Here's the inner membrane. So this is the lipid bilayer. And then not shown here is the outer membrane. So here where it says intermembrane space, here's the outer membrane that's not shown in this picture. So this is focusing exclusively on the inner membrane of the mitochondria. Notice all these integral proteins. So you have complex 1 up here at the top in green. That is an inner membrane protein, integral protein. You have complex 2. You have complex 3, complex 4, and then you have complex 5, which is really the ATP synthase. That's going to be the protein that ultimately makes ATP. Okay, a couple of key ideas then is that each of these complexes will contain a series of redox reactions. And we're going to look at each of the complexes separately for the next several slides. Okay, but that's our overarching idea is that they have a series of redox reactions. Redox reactions, because they are electron transfers, will generate a flow of electrons through this inner membrane. So you see this yellow part here with the E. That means that's going to be our current that runs through the inner membrane. The voltage on that is actually around 1 volt. So that's pretty good, and we'll talk about how that translates into energy. Okay, now where does that energy go? What's the point of a battery is to generate energy to do something. And the energy goes to helping push H plus from the matrix into the intermembrane space. So you're going to have within the intermembrane space, you're going to have a buildup of H plus acid. And it's going against its gradient, and that requires energy. Out of the 4 complexes, complex 1, 2, 3, and 4, 3 of them are proton pumps. So complex 1, see how there's an H plus shown here, pumps it out. Complex 3 pumps H plus in there, and complex 4 does as well. So those 3 complexes push H plus into that inner membrane space, not shown as the outer membrane. That requires energy because they're pushing the H plus against its gradient. And that's the whole role of this energy from the 1.10 volts, the battery, is to help push those protons out. And then what's that going to do is ultimately that H plus is going to flow through this incredible ATP synthase, and that rush of H plus back through that enzyme is going to phosphorylate ADP to ATP. Okay, so that's kind of an overview. And then one other thing that I think is so important is that our couple things, see where it says NADH shuttled from the cytosol, meaning that the NADH, that's from glycolysis. That's what has to get shuttled in because it has to get into the membrane. Into the mitochondria and into the spot where it needs to be. That requires a special carrier. Citric acid cycle occurs already in the matrix of the mitochondria. And interestingly enough, see the step of succinate to fumarate. That's a step in citric acid cycle that actually that enzyme that's involved in that step is actually part of complex 2. So complex 2 is part of citric acid cycle. Okay, there's a lot going on this slide, and it just depends on how much chemistry you have had. But the idea is the following. See where it says E naught here? That's a voltage. And the voltage for this whole process is around 1.10 volts. There is an equation in chemistry that will determine how much free energy is given off by that voltage. Now, if you look, the equation and concept is the bigger the voltage of that battery, the more energy you're going to get out of it. So this has a voltage of 1.10 volts. So the free energy for that is shown down here at negative 220 kilojoules per mole. So it's very exergonic overall, and that is helping to drive the formation of ATP. Okay, so let's go to the next slide. These are what are called standard reduction potentials. These show the ability of a species to gain electrons. And I'm going to click to the next slide. At the bottom of the standard reduction potentials in biochemistry is this super important one. 1/2 oxygen gas plus 2H plus 2 electrons make water. And the potential for that reduction, now why is it a reduction? Gain of electrons. That is plus 0.82 volts. When it has a positive reduction potential, it means it wants to gain electrons. So how do I read these charts? Is the more positive this number is, and see how they're getting more positive as we go down, it means that they want to gain electrons the most. So at the very end of this process, the ultimate electron acceptor is the oxygen we breathe. So the oxygen that we breathe in is going to accept that all the electrons flowing through this process. And if you notice, let me click back to the previous slide. As we begin our process, we're going to start looking at NADH coming in, FADH2 coming into the process. These guys will be coming from glycolysis and Krebs cycle, and they're going to enter into this process. And I might have forgotten FADH2. So remember, from glycolysis, we got 2 NADH. From pyruvate to acetyl-CoA, we got 2 NADH because it turned around. And then from acetyl-CoA in TCA, right, citric acid cycle, we got 6 NADH, right, 3 times 2. We also got 1 FADH2, which I think I didn't mention at our earlier summary, also an electron carrier, and then 2 ATP essentially from 2 GTP that were formed. Okay, and then remember the 6 is because it's 3 times 2 times through. Okay, now what I want to show you that is really important, and there's a nice graphic coming up. These guys have negative potentials for reduction, which means that they really are, compared to ones further down that become positive, they're going to want to be oxidized. So that's how we should think of it. There's a better graphic coming up, is that as we go through and we see all the processes going on, and as we see all the processes in the upcoming slides, we can explain it through these voltages, essentially, that the more positive species will want to gain compared to the negative ones on these charts. So if they're more negative, they're going to want to oxidize. So remember, in redox, something always oxidizes and something has to be reduced. They're processes that go together. And at the end of the day, the oxygen we breathe is the ultimate electron acceptor in this whole process. Okay, here's another picture of complex 1, complex 2, complex 3, and complex 4. Not shown in this pic is ATP synthase, so that's not shown here. What I want you to think about, this is an overview. Here's the matrix. Here's the inner membrane space. Not shown is the outer membrane. Okay, so that should be in your mind that you have an outer membrane. The other thing too is that they're complexes, meaning that there are a bunch of different proteins and they have names. So like complex 1 is NADH coenzyme Q reductase. Complex 2, that aligns itself with citric acid cycle. So this one comes up with citric acid. That's succinate coenzyme Q reductase. Complex 3 is cytochrome C coenzyme Q oxidoreductase. Complex 4 is cytochrome oxidase. Do you need to know those names? No. But you do need to understand that they're called complexes. Now, what are the key players in all of this? Each of the complexes has some very interesting molecules that are involved in electron transfer redox reactions. And so there's a whole wide variety of electron carriers. I kind of think of this as sort of an electron hot potato game. I'm not sure hot potato is even a game anymore, so I might be dating myself with that. But you just should think of it that one carrier grabs the electron, and then it's going to pass it to an electron species that wants the electrons more. So it's always going to flow from an electron carrier with a negative potential. It's going to pass it to one that has a more positive potential. And when it does that, it's because the one with the more positive potential wants the electrons more. It wants to be reduced more. Okay, ultimately, where does this end up? Look down here in this picture, in this graphic. Oxygen, ultimate electron acceptor. It wants to be reduced the most. And so it's going from the one that wants to be oxidized up here, and it's going to pass the electrons, keeps passing to ones that want to be reduced more until it gets to oxygen, which wants to be reduced the most on this incredible list of electron carriers. Okay, now, who are the electron carriers? There's ones called flavoproteins. They contain FMN or FAD. So remember our friend FAD. You saw a picture of it in the previous PowerPoint. It's going to have iron sulfur proteins. Okay, so for example, FES is going to be, and I'll have some pictures of that, and also this very unusual one here. It's going to have a very famous species called, or molecule called coenzyme Q, also known as ubiquinone. And then it's going to have what are called cytochromes, which contain heme. So remember our friend heme from hemoglobin? And so there's different kinds of cytochromes, B, C, or A. And so those are kind of our key players. And these different types of electron carriers are embedded within those complexes. So this is just showing you some pictures of these guys, structures of iron sulfur clusters. So this is a iron, there's the protein as part of the complex. Here's our friend cysteine, the amino acid cysteine, and it's where the sulfur, right, comes from. And then they are bound to the iron. This would be our Lewis acid base type chemistry, where the cysteine donates its electrons to the iron as a charged ion. Here's the structure of coenzyme Q, and it is a lipophilic, meaning that very nonpolar. Do you see it? And I think that that's a really important distinction for coenzyme Q, because you're going to see that it's going to have to, it's embedded and flows through the cell membrane bilayer. So it's going to want to complement the lipid bilayer. It can transfer 2 electrons in one electron steps. So 1 electron at a time. Okay. Now that's the kind of detail that you don't need to know, but I just want you to see, we're going to be a little bit thorough here. So here's the oxidized form of coenzyme Q, and it gains 1 electron to go to semiquinone form, and then another electron to form the fully reduced form. Cytochromes. You should be excited to see heme again. Okay, so the cytochromes contain heme groups attached to the protein. Okay, and so here's the protein, and it's attaching through this sulfur, right? The amino acid cysteine helps, latches on to the heme group. Here's cytochrome C, cytochrome C1, that's its structure. Here's cytochrome A and A3 heme. They are classified according to their absorption of light. And so there's the chart that shows how they absorb light differently. And all right, so the cytochromes are definitely, their structure should look a little bit familiar because of the heme group that we saw in hemoglobin. Again, reinforcing the idea that iron is a very important mineral not only for oxygen carrying, but also for this particular process. Okay, I like this picture. You don't need to know it in detail. You don't need to know the numbers, but what it's trying to show is that our NADH will come in as our electron carrier, and its potential is that it wants to oxidize compared to the rest of them. So it's going to oxidize, it's going to lose its electrons. And those electrons start to get transferred. So here's the hot potato business, right? Boom, it keeps moving and moving through the inner membrane. So you have that flow of electrons generates a current and ultimately will go to oxygen. That is our ultimate acceptor of the electrons, and it's going to reduce oxygen to make water. So that is the final step. Is it's going to make H2O. Okay, so that is the final reaction. Okay, and I like this because what it's showing you is sort of that flow based on the voltages. And so over on this side are the voltages. And then remember, the electrons are going to flow towards species with the more positive voltage. And then as the electrons get transferred, you see how the free energy gets released. So each of these steps releases free energy, and ultimately, across the board, you get a release of negative 221 kilojoules per mole, which is a lot, actually. And again, the voltage is 1.10 volts. Okay. This points out that this chart's based on standard conditions, and in the cells may not be standard, but still, it's a good way of visualizing what's happening in terms of free energy. Okay. Here, I kind of like this picture because it just identifies within each complex what are the important key players. So within complex 1, NADH comes in and it passes electrons to FMN, flavin mononucleotide, to an iron sulfur. Not shown here is that there's a proton pump. The electrons then get transferred to coenzyme Q. In complex 2, that comes in from Krebs cycle, FAD H2, right, is formed from Krebs. That is going to send electrons through the iron sulfur centers to a cytochrome, ultimately reducing coenzyme Q. Coenzyme Q then goes through the lipid bilayer. There's motion there, and it will go to complex 3, which is a series of cytochromes. Cytochrome B, iron sulfur, cytochrome C1. You see these numbers here, 562, that has to do with the absorption of light. You know how they're categorized according to their absorption of light. Then you have ultimately those electrons will end up at cytochrome C. Cytochrome C goes through complex 4, which is a series of other cytochromes, ultimately reducing oxygen to water. Now, do I ask you to memorize the order here? No, but you should know that the key players are iron sulfur centers. You should know that cytochromes are involved. You should understand that you have redox reactions. You should understand the voltage that's generated. So you don't need to know, like, I won't ask you which complex is cytochrome B 562. I won't ask that kind of detail, but certainly I could ask you what's the role of the cytochromes. They are electron carriers. So it's sort of the conceptual piece, not so much the specifics of which complex has what in it. Okay. Also important would be to note, and it's going to come up later, complex 1 will have the NADH. Complex 2, that one's linked to Krebs cycle. That has the FADH2. Okay, and because FADH2 comes in complex 2, as its electrons flow through here, it won't have as much energy to give, and we'll talk about that later. So in other words, you're going to get less ATP from FADH2 because it comes in later in the electron transport chain. Okay, but hold that thought. Okay, so we'll briefly go through each of the complexes. These have here, I like this picture, it just keeps it simple. NADH comes in. So this is the NADH from glycolysis, from the pyruvate to acetyl-CoA, from Krebs cycle. The NADH comes in. It's carrying electrons from those processes. It is going to transfer electrons. Now it's going to give its electrons to FMN, an electron carrier. Now it goes from oxidized to reduced. It's going to transfer it to those iron sulfur centers. Over here, this is oxidized. This guy's reduced. Electrons flowing. And then ultimately, coenzyme Q goes from oxidized to reduced. Okay, so that's the electron carrier process. Now, the specifics that I'm not going to go into is that some of them accept one electron at a time, some two. But what I want you to realize is that, so that part, you don't need to distinguish between 1 and 2 electron carriers. But what I want you to understand is that as a result of all those electron transfers, at points along that exchange, that H plus is given off, and that's pumped. See where it says here? From the matrix of the mitochondria into the inner membrane space. So this is coupled to the H plus pump. So acid is being pumped. And that is a really important piece, and it's pumped against its gradient. Here's a really incredible picture. I hope that this is something that kind of makes worthwhile all the effort you've put in up until now in the class, that you're looking at this and you're saying, wow, there's the lipid bilayer, there's the integral proteins, alpha helices, there's the really interesting part of the complex that has the FMN, the iron sulfur clusters. NADH is going to come in. And as a part of that redox, we have our overarching concept that as those electrons flow, things change in the protein, right? Things move, and there's a conformational change that pumps H plus from the matrix into the intermembrane space. Not shown is the outer membrane here. And you're increasing the H plus, the acid level in the inner membrane space. And then ultimately, complex 1 will form reduced ubiquinone or coenzyme. Ubiquinone and coenzyme Q are the same thing. And that's the reduced form. Okay, here's the another look at it. This shows the conformational change that occurred, and that pump. There's a motion, right? Like a mechanical movement. So the current actually has a mechanical component to it. It's actually allowing for that conformational change that allows for the pushing of H plus from the matrix into the intermembrane space. Here's the second one, complex 2, succinate coenzyme Q reductase. It's accepting electrons from FADH2 or to make FADH2. So this is the idea of it being linked to step 6 in citric acid cycle. So succinate dehydrogenase was one of those enzymes, and it uses FAD to extract electrons from succinate. Okay, and then there's a series of redox reactions here as well, and ultimately reducing coenzyme Q. There is not a proton pump in this particular complex. Okay, so we saw that coenzyme Q, hold up, collects electron from multiple flavoproteins for transfer to complex 3. Okay, so both complex 1 and complex 2 take ultimately reduce coenzyme Q. And now coenzyme Q will go through the membrane and enter into complex 3. So electrons from reduced coenzyme Q pass to complex 3. Okay, so complex 3 will catalyze the transfers from reduced coenzyme Q to cytochrome C in the intermembrane space. And so this is showing you kind of, it's really incredible looking structure. And so you have a series of cytochromes within there. Here's another one showing all the different types of redox reactions that are occurring. So you don't need to know the specifics here, right? Except to understand that there's a series of redox reactions. It involves some cytochromes, some iron clusters, and ultimately, it's going to take the reduced coenzyme Q, and ultimately what it's going to do is it's going to reduce cytochrome C. So is reduced. There's also, if you notice, this is also a proton pump. So the net process pumps 4 protons into the intermembrane space. Okay, now very complex, as you saw from the previous pictures, and this might scare you. So just highlighted the 2 main ideas. That coenzyme Q that's reduced, that came in from complex 1 and 2, is now going to ultimately transfer its electrons through a series of cytochromes, ultimately reducing cytochrome C. At the same time, it's pumping more H+, more acid into the intermembrane space. Okay, where does this all end up is, so the reduced cytochrome C comes now to complex 4, and the ultimate goal in complex 4 is that the cytochrome that's reduced goes through a series of electron transfers and ultimately will reduce the oxygen we breathe to water. Also a proton pump as well. So that's the important piece, is that all the series of redox leads to the reduction of oxygen, and at the same time, it allows for the pumping of H+ against its gradient in the intermembrane space. I also think that it gives us a really great appreciation for all the amazing secondary structures and just the whole look of these integral proteins, right? And I hope that you're making connections to why we learned about the cell membrane, what it was like, why we tried to think and understand protein structure, because it's really important here as we look at these pictures. Okay, so the free energy ultimately is negative 220 kilojoules per mole. That's from a voltage of 1.10 volts. And what is going on here is the idea that NADH comes in complex 1, and it's going to produce around 2.5 ATP for every one NADH. Since FADH2 comes in complex 2, it's going to produce 1.5 ATP. So it might sound funny, the 2.5, 1.5. If we think of it more as a mole quantity, then the halves make a lot more sense. And the electron transfer reactions are not directly coupled to ATP synthesis, as we'll see. So that's why they're not integer values. But these will be important numbers for us to consider when we look at our final bookkeeping. Okay, this slide isn't so important. Okay, our last section is on the chemiosmotic coupling and ATP synthesis. So I like this slide. It's more simple. It shows the NADH in complex 1, proton pump, coenzyme Q transferring electrons to in the process. Here is complex. Oh, is complex 2 even shown here? No, it's not shown. Oh, I know why it's not shown, because it's not a proton pump. So it's only showing proton pumps. Here's complex 3, and here's complex 4. Here's the important reduction of oxygen to make water. So if you notice, here's the flow of electrons, that current, right, flowing through. What is happening is that then you have a high concentration of acid on this side in the intermembrane space. And there's this idea of what happens next, and it's called the chemiosmotic coupling method, that the electron transport drives the transport of protons, so that's the proton pump, and it creates a gradient where you have high concentration of acid, and that's a charged species. So why electrochemical gradient? Because it's charged. It's a charged, so that's where the electro part, and chemical acid. And so you have a high amount of H plus, and what's going to happen is it's going to flow through ATP synthase. Here's the ATP synthase, complex 5, and that flow back through that protein is going to phosphorylate ADP to ATP. Now, the ATP synthase is a very complicated protein. I always marvel at it. Again, this going to have a more detail than you need. But what I want you to think about is what an amazing protein it is. It's going to be an enzyme, and it's going to catalyze the phosphorylation of ADP to ATP. It's a proton pump because the H plus is going to flow through it. And here's what's really amazing is it has a very distinctive rotation, a physical rotation it does that's a lot like a molecular motor. So that's pretty neat. So let's take a look. It has ATP synthase. So here's the name of it. It has two complexes, F1 and F0. F1 is the site of ATP synthesis, and F0 is an integral membrane protein that's attached to F1. So here it is in all its glory, the picture. See, here's the F0, right, in this part here. And then you have F1 is essentially, if you look at it, it looks like a knob, right? And it's the one that's going to rotate. It's literally going to rotate like a knob. Okay. What an incredible looking protein, correct? It has a part that's going to allow, right, for the flow of H plus through it. And then as H plus flows through it, that bottom part, the F1s will start to rotate. And it's actually that physical movement that's going to help phosphorylate ADP to ATP. Now, what I just verbally said is really the important part. I'm not going to ask you for the nitty gritty on this. I am hoping that you look at this and think, oh my gosh, what an incredible protein. But I'm not going to ask like specifically the different subunits of it. Okay, so let's take a look. Next one. There's the F1. There's the F1 complex, kind of has the doorknob looking, right? Complex that rotates. This is optional, if you want more detail about that motion. Essentially, what happens is that the F1, the knob part, has three different conformations. L, which is loose, T tight, O is open, and then there's a rotation. And so ADP actually comes in, and as it rotates, that rotational motion starts to essentially create almost a forcing a phosphate onto ADP. Okay. So there's sort of this actual movement of that, like a knob, that creates kind of almost like a whipping motion that's going to phosphorylate ADP to ATP. If you want to read more carefully this for more details, you can, but I don't want to make it seem like you're going to have to describe it. But it obviously has a lot of different subunits involved in that whole process. So we're just looking at it more on an introductory level. I do like this last sentence though here. Conformational changes allow for the binding of ADP and phosphate to form ATP. Okay, so you see that in this case, the electrochemical gradient, the flow of protons, so H plus. Now, in all our other pictures, the inner membrane was on the bottom side of the slide, so this is flipped up. So here's our inner membrane space, outer membrane not shown. And so the H plus is going to flow through the top part, the FO, right? This part here. And as it flows through, it's going to help to create that mechanical motion that's going to phosphorylate ADP into ATP. Okay, and so it's that flow. Think of like a waterfall that has water that's flowing down. That creates a huge amount of energy, right, to go from high to low. Same type of idea here. The high concentration of acid on this side goes through that protein, and then that is going to allow and provide the impetus for that rotational motion in the F1 unit. Okay, so what an incredible process. I find it so fascinating, and I hope you do too. And I hope you don't let all the details bog you down, because really, ultimately, you're seeing from NADH and FADH2 a series of redox reactions that creates free energy that pumps protons from matrix to intermembrane space. Those protons flow through ATP synthetase to create that rotational movement in the F1 subunit that phosphorylates ADP to ATP. And in the process, those electron carrier reactions reduce oxygen to water. Okay, so here's our energy yields. There's a lot more ATP generated from oxidative phosphorylation than was seen in substrate level phosphorylation. So from glucose, we have 2 NADH. From pyruvate dehydrogenase complex, that was pyruvate to acetyl-CoA, 2 NADH. From citric acid cycle, churning around twice, 6 NADH, 2 FADH2, and here's 2 ATP. So those guys are done. You know, those don't need to be accounted for because they're already ATP. So ultimately, let's add them up. 2, 4, 10 NADH, 2 FADH2, 4 ATP, because glycolysis had 2 ATP plus those 2, right? And then let's think about our conversion factors. So you have for every 1 NADH, you have 2.5 ATP. For every 1 FADH2, you have 1.5 ATP. So if we do all of our math, what we would get, see where it says using the ratio of 2.5 for NADH and 1.5 for FADH2, we're going to calculate the total yield of 32 moles of ATP per mole of glucose oxidized. Okay, and I think that this is an important discussion to kind of see the big picture because we get so much more ATP from oxidative phosphorylation than we did from glycolysis or from citric acid that was substrate level phosphorylation. And then for some of our organs, there's an energy toll bringing in NADH, shuttling it into the mitochondria. So those of you that might have learned this in another class, in a bio class, they usually give it as a range of 30 to 32 ATPs for one glucose, because there's shuttles that have to bring in NADH, and so there's an energy cost to that. And here are the shuttles. I'm not going to ask questions on these, but you have this PowerPoint, so you can go ahead and read a little bit more. So again, energy cost to moving NADH in from the cytosol into the mitochondria.